Good morning. Today I'm going to talk about macromolecules. Macromolecules are very large molecules such as proteins, carbohydrates, lipids, proteins, etc. It is formed by the polymerization of smaller subunits. It is typically composed of smaller subunits which are joined together to form a macromolecule. A macromolecule, the term was coined by Hermann Staudinger in 1920. There are four major classes of biological macromolecules that we come across day to day, that is carbohydrate, lipids, proteins and nucleic acids. So we're going to move into the first category known as carbohydrates. Carbohydrates are divided into four categories: monosaccharide, disaccharides, oligosaccharides and polysaccharides. So now we're going to take up to monosaccharides. So carbohydrates are divided into four categories: monosaccharides, disaccharides, oligosaccharides and polysaccharides. Monosaccharides. Monosaccharides, as it indicates, mono stands for one. So monosaccharides has got one carbon atom, that is one carbohydrate linked together. So it's known as monosaccharides. The typical example for monosaccharides we come across day to day are carbohydrates. There are four categories of carbohydrates: monosaccharide, disaccharide, oligosaccharide and polysaccharides. What are carbohydrates? Carbohydrates are nothing but sugars. They are broken down during a process of metabolism to release energy. So they are different categories. Under that first category is monosaccharides. So monosaccharides are like one sugar, and that is known as simple sugar. As the name indicates, disugar, 2, 2, 3, many sugars. So under monosaccharides, the first examples what we are gonna see is aldehyde groups and ketone group. If carbon atom is the last compound and this is the last chain with one side chain, that is one R side chain, and if a sugar, that is a monosaccharide, has this group C, last on the group with a single R side chain is termed as aldehyde groups. This is termed as aldose sugar. This is aldose sugar, which you come for major examples for monosaccharide. Next, under for ketone group, where C is the central or internal, C is internal on the chain, meaning there are carbon on both sides. Then it is termed as keto sugar. It indicates that there are possibilities of two carbon atoms accompanying here, but the possibility when a side chain of C carbon is at last, there is a possibility of only one carbon chain accompanying here. So this makes the difference for monosaccharides. Next, we are moving on to disaccharides. As the name indicates, di for 2. So there are possibilities of two sugars joined together by glycosidic linkages. Formed by the removal of, actually the disaccharide is formed by the removal of hydrogen atom from one monomer and an hydroxyl atom from another monomer combined together to form a disaccharides. So commonly the examples for disaccharides are maltose, sucrose, etc. So what an example like we are going to see here is maltose. So maltose is formed by the combination of glucose plus glucose. So two monomers, this is single monomer plus the second monomer joined together to produce your disaccharides. The monomer, what happens is here, hydrogen from this monomer and hydroxyl from another monomer joined together, and so the byproduct along with this will be water. So this is an example for maltose. Next comes sucrose. For sucrose, it is glucose plus fructose. Next is lactose, which is presence of galactose plus glucose. So this is around carbohydrates. So main examples monosaccharide, disaccharide, oligosaccharides and polysaccharides. So we now seen upon monosaccharides and disaccharides. Next we are moving to the third category of the group carbohydrates. The third category is polysaccharides. Polysaccharides. The main function of polysaccharides is storage and structure. So the main function is storage and structure. The sugars which are involved in storage are starch and glycogen. And for structure, it is cellulose and chitin. So starch are nothing but sugar. It is like a glucose alpha configuration is termed as starch. Starch is usually prepared during photosynthesis by plants and it is stored in plastids. What happens to the starch is the starch is utilized by the plants and it is broken down during a process to release energy. Next, when you come across glycogen is present usually in liver muscles of animals, higher order animals. What happens here is glycogen phosphorylase enzyme is capable of breaking down glycogen to release energy to the muscles. Next comes cellulose. Cellulose is the most abundant inorganic compound which is present in almost all the cell walls. The cellulose is broken by cellulase enzyme. Enzyme is capable of breaking only alpha glycosidic linkages, but it is not capable of breaking beta glycosidic linkages. And another example is chitin for structure. Chitin is present in the exoskeleton of all the cell walls and it has been hardened by addition of calcium carbonate to the cell wall. The main characteristics of chitin is it is flexible, it is slippery, flexible and slippery. Now these are the main functions of polysaccharide. So we have talked about storage and structure. Under storage, starch and glycogen. So starch is usually made up of amylopectin. For glycogen, the enzyme which is used to break glycogen is glycogen phosphorylase. Cellulase by cellulase enzyme. And finally, we have chitin, which is an exoskeleton which is hardened by calcium carbonate addition. Now, we are going to move on to the second category of macromolecules. After carbohydrate, we are moving on to proteins. So, the building block of proteins are amino acids. So, proteins are made up of 21 different amino acids. The main structural prediction of amino acids are containing 5 structural formulas which are structural groups which are combined along with the amino acids to give a structural stability for the protein. So they are the first one is carbon. The second one is H atom, that is hydrogen atom, and the third one is amino group, and the fourth one is carboxyl group, and the fifth one is R side chain. So the formation of the group will be like this: H, N, H, H, which is nothing but your amino group, followed by C, H and R, that is your side chains which is incorporated along with your major structural. Then next comes the carboxyl group. So this gives you this is your amino group, this is your R side chain, this is your carboxyl group. So this is the main structure of your protein. Along with this you have your amino acid, but they also have a carbon, H atom, amino, carboxyl and R side chain. And there are major types of proteins. They are non-polar, major types of proteins. First types is non-polar. Non-polar are hydrophobic. So they are hydrophobic and they lack any type of changes They lack any type of changes capable of forming H bonds. Capable of forming H bonds. Next comes the second group, non-polar. Next comes the polar group. Polar group are hydrophilic in nature and they have partial changes forming H bond. Under H bond you have delta minus and delta plus categories. Next, third category comes the charged. So when you take COOH, the COO minus, and when it comes for amino group, this NH, any category, NH2, NH1, NH3, any category of amino group, so this NH and CO minus are imparting ionic strength through ionic bonds. So this is considered as basic charge and this is considered as acidic. So based upon the polymerization, it is divided into, based on polymerization, it is of two types. The first type is monomer and next comes the polymer, which you have already seen, one compound and for two compounds, 1, 2 or 3. So when a monomer is combined, like I already explained, monomer, this hydrogen atom links with the OH of the other monomer. Here also you have a hydrogen, so the linking takes place and it produces a monomer plus monomer, that is a polymer. And byproduct you will have your water. So there is a peptide bond linking between a protein and a protein, peptide bond. That is N terminus, that is amino terminus, and you have your C terminus, that is carboxyl terminus. So for N terminus I am drawing amino group. So this terminus, this is a sequence of your compound, sequence of your amino acids joined together on both sides by amino group and a carboxyl group, and this sequence or this bond between these amino acids are termed as peptide bond or covalent bond. The main properties of proteins are solubility. Like if they tend to form in polymers, it is tend to form colloidal nature. So insolubilities and solubility is also seen in proteins. Denaturation, elasticity, gel formation and maillard formation. So now under carbohydrates, like we have seen few categories, like under macromolecules, carbohydrates, under carbohydrates, monosaccharide, disaccharide, polysaccharides, and after that you have gone to the proteins. Under proteins, different types of proteins based upon their polarity and charge and based upon their polymerization, monomer and polymer, and their sequence analysis. Now we are going to move on to the third category of macromolecules known as lipids. So lipids are commonly hydrophobic in nature. Lipids are commonly hydrophobic in nature. So you have a major category of lipids are known as fatty acids. Under fatty acids you have saturated fatty acids and unsaturated fatty acids. This is commonly seen lipid varieties which we come across day-to-day life, but there are also some other types of lipids which I am going to teach you now. So under lipids, the main category, as I said, fatty acids and non-fatty acids. So under fatty acids, you have saturated fatty acid and unsaturated fatty acid. And the second category comes glycerides. So under glycerides you have neutral glycerides and phosphoglycerides. You also have a non-glyceride category, non-glyceride lipids. Under non-glyceride lipids you have waxes, sphingolipids and steroids. So under sphingolipids you have sphingomyelin and glycolipids. There is also one more category of lipids which we don't come across, and they are known as complex lipids. The main examples are lipoprotein. So these are the main divisions of lipids. Under lipids you have fatty acids, glycerides, non-glyceride lipids. Under fatty acids you have saturated fatty acids, unsaturated fatty acids. Under glycerides you have neutral glycerides and phosphoglycerides. Under non-glyceride lipids there are three categories: waxes, sphingolipids and steroids. Sphingolipids are further divided into sphingomyelin and glycolipids. One of the rare category of a lipid is complex lipid, and they are known as lipoprotein. So now we're going to see structure for every compounds. So first starting with saturated fatty acids. Saturated fatty acids. One of the best example for saturated fatty acid is butyric acid. Next is unsaturated fatty acid. Unsaturated fatty acids. The best examples will be triglycerides. The main difference between saturated and unsaturated fatty acids are, saturated fatty acids are nothing but it is in single side chain, and there are no complicated bonds which are formed in saturated fatty acids. But when you come across unsaturated fatty acids, they are complicated and they have more side chains than compared to the saturated fatty acids. So example for saturated fatty acids is butyric acid. Like if you see, you can have a COOH group, CH2, CH2 and a CH3, butyric acid. You can all see there are all only single bonds. So when you come across unsaturated fatty acids, you can see there are double bond, and the number of the side chains, that is R1, R2 and R3, the functional side chains are more when compared to the saturated fatty acids. You also come across phospholipids. Phospholipids are the major component of a cell wall. So when I said it is hydrophobic in nature, this phospholipids also have a hydrophilic tail which has been combined with the cell wall. So phospholipid structures are like this. So this is hydrophobic. You call it as hydrophobic head for phospholipid, and these are hydrophilic tail. So this is hydrophilic tail. So if you take a single structure and if you see it, you can see that this is the hydrophobic head, that is known as a phosphate group. This is the phosphate group which is present here, and you also have a two head-like structures which is seen under your phosphate group, and this is nothing but glycerol. So this is a detailed structure of a single phospholipid, and you got a two tail, and this tail is nothing but two fatty acids. So this is a typical structure of a phospholipid. So now from the morning onwards, we have seen the macromolecules, the major differences of the macromolecules such as carbohydrates, proteins, lipids. Next comes the fourth category, it is known as nucleic acids. Nucleic acids are the main component of a living organism to carry genetic information from one organism throughout the body, from one generation to another generation. There are two types of nucleic acids. One is known as DNA and another one is known as RNA. DNA, as the name indicates, it is deoxyribonucleic acid, and RNA is ribonucleic acid. Deoxyribonucleic acid, the main difference is it is made up of deoxyribose, whereas RNA it is made up of ribose sugar. DNA and RNA plays a very vital role in each human being because it is the part where your genetic information is stored and it has been carried. Whereas in RNA, here it happens coding and non-coding of genes takes place. For a protein to be expressed, RNA should be present. Whereas DNA is a permanent part of all living organisms. RNA seems as temporary and it is also not stable. So now we're going to look at the structure of DNA as well as RNA. So nucleic acids, this is a fourth category of your macromolecules, biological macromolecules. So nucleic acid structure, if you take, you have DNA and RNA. So DNA is nothing but deoxyribonucleic acid. Deoxy for deoxyribonucleic acid. And if you take RNA, it is ribonucleic acid. Ribonucleic acid. So when it, actually if I said a macromolecules are nothing but large number of monomers are joined together to make a polymer. So nucleic acids are commonly done by joining of nucleotides. So number of nucleotides joined together along with the sugar and a phosphate group. So there are three main components in a nucleic acid. They are sugar, phosphate group and nitrogenous bases. So first one is sugar. So I already mentioned deoxyribose sugar. And another one is ribose sugar. Ribose sugar. Deoxyribose sugar, it is seen in DNA and ribose sugar is seen in RNA. These sugars are joined together by phosphate groups. Next comes the second category of your nucleic acids or second component of your nucleic acid known as phosphate group. The phosphate group is formed by an inorganic compound phosphoric acid which is getting combined with your nitrogenous bases and sugar, and this is a building block. So next third category, so phosphate group is made up of inorganic phosphoric acid. So it is nothing but H3PO4. Next comes your nitrogenous bases. Nitrogen bases are divided into two categories, purine and pyrimidine. If you take purines, they are made up of adenine and guanine. If you take pyrimidine, it is made up of cytosine and thymine. So purine and pyrimidine. So adenine, guanine, cytosine, thymine are the nitrogenous bases which are seen in the nucleic acid. So there are three main components seen in nucleic acids. They are phosphate, sugars and nitrogenous bases. So they are joined together to form a double helical structure. So DNA will have a double helical structure whereas RNA will have a single helical structure. This double helical structure is wound around each other in a right-handed fashion and they are anti-parallel. Whereas in RNA, it is a single helical structure. And the main difference between DNA and RNA are not only in their sugar formation but also in their nitrogenous bases. If you come across DNA, it is made up of thymine, a nitrogenous bases, but it is been replaced in the RNA by uracil. So for RNA, it is adenine and uracil. And adenine, uracil, guanine and cytosine. Adenine, guanine, cytosine, thymine and uracil are the 5 nitrogenous bases which are found together in a nucleic acid. So when we divide from DNA to RNA, the nitrogenous bases sequence change. For example, adenine, guanine, cytosine and thymine are found in DNA, whereas adenine, uracil, cytosine and guanine are found in RNA. So there are also formation of purine and pyrimidine. So purine, under purine you have adenine and guanine, and cytosine and thymine comes under pyrimidine. Adenine and thymine are joined together by 2 hydrogen bonds, and whereas cytosine and guanine are joined by 3 hydrogen bonds. So adenine and thymine are the nitrogenous bases which are joined by 2 weak hydrogen bonds. Whereas cytosine and guanine are joined by 3 weak hydrogen bonds. So if you take a typical structure of a DNA, it will be like For example, first for thymine, this is for adenine. So this is the structure of thymine and adenine. So if you see, you can see I say two hydrogen bonds. It is made up joined of two adenine and thymine are made up of two hydrogen bonds. So these are the hydrogen bonds. You can see. And for purine, it is made up of adenine and guanine. The purine is capable of forming two rings, whereas pyrimidine will have only single ring. So if you see, you can see two ring formations here, indicating that that is of an adenine, whereas a thymine you have only single ring. So under pyrimidine you will have thymine and guanine. So this is all the major chemical structure formula of your DNA. So now when we go on to cytosine and thymine, cytosine and guanine. Under cytosine, so this is the structure of cytosine and guanine. In cytosines, as I already said, cytosine and guanine are linked by three weak hydrogen bonds. So you can see one hydrogen bond here, second hydrogen bond and third hydrogen bond. And cytosine comes under the category of purine. So under cytosines come under the category of pyrimidine. Comes under the category of pyrimidine. So pyrimidine will have only one ring structure. So you can only see one ring here. But when you see for the purine, which has been made up of guanine, so guanine will have two ring structures. So you can see guanine is made up of two ring structures. So these are the chemical structures of DNA. Now we are going to move on to the ribonucleic acids. Ribonucleic acids, which is made up of, instead of thymine you will have a uracil compound.